already know how this works?
Yeah.
Yeah.
Do we already know how these work?
Yeah.
I'm only adding like 3 new things. So, what we learned before, gonna be similar today. Alright, electron carriers. What's carrying our high energy electrons? NADH. Look back through your notes. With glycolysis and Krebs, how many NADH did we make in total? 8 in Krebs.
10. Yeah, 10.
And 2 in glycolysis. So
10.
10. So our NADH, because it's carrying our high energy electrons, it's going to drop off electrons at the first hydrogen ion pump. Because those electrons now have enough energy to travel through the entire chain and power every pump before they lose all their energy. So from beginning to end, they're just losing a little as they go. Our 2-FADH2 is carrying what? What's this carrying? Low energy. If they dropped off at the beginning and they're low energy already, would they have enough to make it to the end? No. So they get dropped off further down the line. Okay, so they're going to get dropped off here and enter the ETC at the second pump. So they only have enough energy to power 2. High energy has enough to power 3. So when NADH drops off its electrons, it's also dropping off hydrogens. How many? Each NADH has how many hydrogens? 1, right? Only 1. So there's going to be 10 hydrogens to get dropped off. FADH2, when it drops off its electrons, how many hydrogens did we say each one had? 2. So it's going to drop off all those. So we're going to have some hydrogens in the matrix, but they're going to get immediately pumped across. Okay, so as soon as electrons start traveling, they're going to power the pump, and it's going to pump hydrogens across. So the hydrogens over here, that's an H. And what's going to happen is we're going to get this high concentration of hydrogen ions in the intermembrane space. I don't care how many you put, but there's a lot. Right? So that's why we need the energy from the electrons, because we're going low to high. Pumping them against the gradient. If you had to take a guess, an educated guess from your prior knowledge, is the pH of the intermembrane basic or acidic? Why would it be acidic?
Because there's more H+.
Because there's lots and lots of hydrogen ions. Okay, so it's going to be more acidic than the matrix. Good job, guys. Okay, so now we have this buildup of all these hydrogen ions. How did they work in photosynthesis? What did they do? They went through HB synthase. And as each one went through, what happened to HB synthase? It turns. When it turns, what does it suck in? ADP and phosphate. And what are we going to make? ATP. Okay, if we have all this buildup here, we pumped all these hydrogens across, right? And they're flowing through, facilitated diffusion here. And we're making HP. We're going to make anywhere between 32 and 34. So as soon as a hydrogen ion makes it back into the matrix, it can go right back through. And just like a little cycle within a cycle. Gets reused. We have talked about glucose. We have not talked about oxygen. This is an aerobic process. Requires oxygen. And here's why. At this last hydrogen ion pump, oxygen is waiting. How many?
6.
6. So oxygen is waiting there. I want you to write this down. Oxygen has an official name in this process. It's called the final electron. Final electron acceptor with an A. Without oxygen here as the final electron acceptor, the electrons would not move across. So if there's no oxygen, this cannot take place at all. No matter how many NADH get made, no matter how many FADH2 get made, if there's no oxygen, nada. And if there's no electrons getting pulled across, are there going to be any hydrogens in here? Is there going to be any HP made? No. So oxygen, super important. So it's waiting at the end. It's pulling all these electrons towards it. And so it's going to pick up electrons. So oxygen's going to come in, pick up electrons. And it's going to pick up any random hydrogens. And it's going to produce water. Six of them to be exact. That's where the H2O comes from. Are you ready to have your mind blown? Yes?
Yes.
Okay. You told me when we talked about glycolysis in animal cells, glucose comes from our food, right? And photosynthesis, it comes from the carbon cycle. So think back to photosynthesis. We talked about tracing the pathway of electrons in photosynthesis. Where did the electrons start in photosynthesis? H2O. We start with water. And then what picks up those electrons in the LDR? At the end. NAD plus, right? So it would go from water to NADPH. And then where does that NADPH go? Calvin, so that we can make what? Glucose. So from water to an electron carrier into glucose, right? And now we have glycolysis taking that glucose, and we're going to break it down. Where do those electrons go then? What's the carrier in glycolysis? NADH, right? Or we make NADH. Then NADH comes here, drops off electrons. What's the final thing to pick up electrons? Oxygen. And what's the final thing we make? Water. So in photosynthesis and cellular respiration in plants, electrons start in water and they end in mind blown. So all electrons in this start with glucose, go to NADH, come to ETC, get picked up by oxygen, and we make water. Are you ready for a final explanation and a final popcorn? Yeah, me too. Alright, so we're going to have NADH from our other processes and FADH2 come to the inner membrane, where NADH is going to drop off high energy electrons. They're going to enter the ETC and start traveling through it and start pumping hydrogen ions against the gradient. FADH2 is going to drop off low energy electrons lower in the pathway. As it travels through, it's going to pump hydrogen ions against the gradient. Those hydrogen ions are going to build up in the intermembrane space. They're going to flow through ATP synthase one at a time, causing it to rotate, sucking in ADP and phosphate, putting them together to make ATP. We're going to get 32 to 34. There's a range because scientists are arrogant and they can't Lastly, oxygen is waiting at the end. Pick up the electrons that have now lost all their energy, along with some hydrogen ions to make water. You got 2 minutes to explain to your C partner.
Everything? Popcorn machine's coming right to you, Tanish. What? The what? And I already did my popcorn. I can't hear you, bro. What is a popcorn? What he tested to you? Well, then you pick somebody else. I mean, you're actually kind of stupid for being smart. Explain to me, man. I don't understand this. Hence, I'm gonna go home and my mom's gonna explain everything. Basically, it's the same as we're gonna have a session on like Monday. LDR process. You have session on Monday. You have your hydrogen ion on Friday. Tomorrow. Exactly. Where? Right, where I can't go. Where? The collection of the hill. You can take my place. You can go up and test the collection. I don't know. Yeah. Where? If like nobody comes. How far is it again? It's not like from us, like 30 minutes. For me, it's like 50 minutes. Oh man, yeah, he was like 10 minutes away from the school. I live like that. Well, it depends on traffic. It's always Normally 15 from there.
If there's no traffic, it's probably like 15 minutes, but with traffic, it takes a lot longer, especially in the afternoon. There's always traffic around that Walmart area.
Explain. What are you doing?
Hold on. So you have your hydrogen ion pump, which is active.
See?
This is the same function, there's a couple different things. And then this, you have the 10 NADH, goes right there. And then you have your pt5 chains, go here.
I'm asking you a shill.
You have two FAD.
You ready?
No, we're not ready.
Ended? I can't remember anything. It was our last person. Oh, pick somebody.
You should pick Tony. You should pick Tony.
No, already gone. Pick
How
somebody.
about Tanish?
I'm already gone. You can't make me suffer again.
Just for that, he gets picked again. Miss Hickson.
What?
Pick Tanish again, just for that comment.
All right, it's
So 10 NADH drops like the electrons.
First of all, where are we? Oh, what's this?
The inner membrane.
Thank you. Okay. So drops off electrons, got it.
And the electrons power they go through the H plus pumps. They power the H plus concentration. The H plus goes through them, and they are like in a high concentration at the top.
Okay.
And
What type of electrons? Did you say?
high power,
High energy.
high energy.
Okay, choose somebody.
Okay, so the high energy electrons are going to travel through the ETC pathway, and then two FADH2 things go to the
Electron carriers.
the electron carriers.
It's not. Smell story.
Okay.
Characters.
So they drop off low energy electrons further down on the pathway, and those electrons are going to travel to the end. And then they also drop off hydrogen ions, which are pumped through the hydrogen pump because of the electron energy.
Perfect.
And they go out. There you go.
So they go off from like a high concentration to low concentration, and so then the oxygen, it picks up the
What goes from?
hydrogen.
Are you doing this?
No, I'm talking about how the hydrogen goes from low to high state.
Okay.
And then so then the oxygen, it picks up the electrons and makes the 6H2O, and so from there, the hydrogen they go to the ATP synthase and they open up, and the ATP in phosphate, they go through and make ATP and they run to ATP34.
Perfect. In total, how many ATP are we getting out of glycolysis, Krebs, and ETC? so 36 to 38 is our range. Questions?
No, not at all.
Can we do that, please? I do want to get checked out.
Alright, so this oxygen, right, it's just a reactant. So it's hanging out in the matrix. So there's all these oxygens there. And it's just pulling electrons towards it. So as soon as one oxygen picks up 2 hydrogen ions and the electrons and makes water, another one comes in. That's why they're very important. Aerobic process. Have you ever been to like the low country that's like salt marshes and stuff?
No.
Anybody been to a salt marsh?
No.
It's got like a rotten egg smell to it. You know what I'm talking about?
No.
Some of us?
Never want to.
I'm gonna go further. You might do some of us. Okay.
Maybe we'll understand if you look at me.
Okay, this sheet is what we're working on the rest of class, however long that is, I don't know what time to get out of here. There is a little bit of fermentation in here. Fermentation is Tuesday. So on Monday, we're gonna draw and practice on the table. Tuesday is fermentation, so 3.4 notes are due on Tuesday.
It doesn't have 3.4 over there.
We still have to learn about fermentation.
It only says
It's easy.
To remembering. Oh, that's like yeast.